All right. So we're on to a whole new unit. You don't want to forget everything we just did because the final is cumulative, but we're kind of starting fresh for this unit. Some of the information that you learned in the first one obviously is applicable here. So we're kind of building. We were talking about atoms and molecules and, well, atoms and molecules basically up until now. Now we're going to get into cells. So kind of one step up that hierarchy that I just talked about. So I kind of like to stop and talk about what we've talked about and then kind of where we're going every once in a while. I think it's helpful. So we talked about how science is done, the organizational structure of biology. So now we're stepping up another level into the cell category. Talked about the basics of biochemistry, which are those biological molecules, the carbohydrates, the lipids, etc. We know water is amazing. It can do a lot of things. Yeah, and then we talked about the 4 biological macromolecules. So those are kind of one and the same. So we'll get into how those macromolecules are used in the next unit. But now we're going to talk about sort of how a cell functions and what makes up a cell. So I think for most of this unit, we're kind of stepping away from chemistry, which is nice. We'll get back into it in the next unit. But for now, we're just going to learn about cells and the different parts of cells and how they function. And I just said all this probably. So we're going to do a tour of the cell, look at the organelles, which are just little tiny organs within the cell. And then the plasma membrane, we're going to focus on specifically. So that's the border that lines the outside of the cell. It's really important in letting material in and out. So we're going to spend, I think there's a whole separate chapter on just the plasma membrane. And then we'll talk about what we call the cell cycle. And that's just the life cycle of a cell. What does it do over its lifetime? A big part of that is going to be cell division, which is mitosis. If you took any kind of bio class in high school, hopefully you've heard the term mitosis. It's just when a cell divides and goes from one cell into 2. So an exact replicate copy of itself. That is mitosis. We'll talk about the steps and how that happens. So yeah, I think that is that'll take up, gosh, a lot of the next few lectures. So we'll get through all of this and it'll be several lectures away, basically, that this will take us through. So there's a lot of material within each of those bullet points, I guess is a way of saying that. But that's an overview of what we're doing. So let's start with cells. Just basic info about cells, kind of some cell background. So in the human body, and we're just one species, but in the human body, there's about 37 trillion cells. So we have a lot of little cells making us function properly. The first cell, they estimate, evolved about 4 billion years ago. So that was just one single-celled organism 4 billion years ago. And there's a lot of different types of cells. So we think of a cell as just this little circle with like a nucleus and some other stuff inside of it. But there's lots of different shapes. There's different functions of cells. This is really just focused on the human body. But muscle cells are different from blood cells. Fat cells are different from bone cells. So there's a lot of differentiation. We're going to talk about cells from just a very kind of generic standpoint, about kind of giving you some background about what's in a cell. And I don't often do history of biology. I think it gets a little bit boring, but the history of cell theory is important to kind of understanding where we got this information. And I'm not going to go into excruciating detail about this, but I'll tell you about a few of the really important players mostly back in like the 16 and 1700s, so a long time ago now. So Robert Hooke was the first person to actually, he named a cell. So before that, no one really knew that there were these little individual pieces that work together to form tissues, et cetera. So he discovered cells. I mean, they were there the whole time, but he discovered them. And his big book was called Micrographia, which he wrote in 1665. And the only cells, well, the cells he started looking at were actually in cork. And cork, you may or may not know, is basically bark tissue of a tree. So it's once living tissue, it's dead now. But he looked at cork under a microscope and the cells were dead, so there wasn't really anything in them. They were just these kind of little circles smushed up against each other. And he named them cells because he thought they looked like the rooms that monks lived in, which are called cells. They live in cells. So that's why we still, to this day, call them cells. So that's Robert Hooke. He's kind of the beginning, the progenitor, I guess, of modern cell theory. And then this guy with a really long, hard to pronounce name came along a few years later, Anton van Leeuwenhoek. And he's considered the father of microbiology. Microbiology is essentially the study of mostly single-celled organisms, anything that you have to look at under a microscope. And the first things he observed were protists. They're just really primitive organisms, and he called them animalcules. So here's some of his drawings of these protists. There's a lot of them. And he was actually the first person to determine that two cells had to come together for sexual reproduction. So a sperm cell and an egg cell had to join in order to create a new organism, a human being, for instance. So they didn't know that before about 1676. So those two guys' names, you want to know their names and sort of what part of the knowledge of cells to attribute to them. So Robert Hooke kind of discovered cells. He was the first one to see cells under a microscope. And then van Leeuwenhoek was father of microbiology and determined that fertilization is required for sexual reproduction. So those are the first two names. There's not many more. I think only, there might be 4 more, but they're grouped in 2s. So formal cell theory came about, or started to come about, in the 1830s. So now we're jumping ahead quite a ways. We were in 1676. So now we're kind of 2 centuries further down the road. People have been studying cells, but the cell theory wasn't really developed until the 1830s by Schleiden and Schwann. Their names are kind of similar, I guess. That's hopefully helpful to remember. So they developed the first 2 parts of the modern cell theory. I'm going to give you 3 different points of the cell theory. And they came up with the first 2, which are here. That is that all living things are composed of at least 1 cell. So if something is made of a cell or more than 1 cell, it's considered alive. And this kind of goes hand in hand with that the cell is the basic unit of life. So if something is not made of cells, then it is not alive. And life is basically built up from that cellular level. All right, so that's Schleiden and Schwann in the 1830s. And then the final tenet of the cell theory, the 3rd one, came along about 20 years later. I have Virchow or Rimac because I can't remember which, but one of them takes the credit, but the other guy actually came up with this 3rd one, and I can't remember which is which, but both of their names are associated with this final point of the cell theory, that all cells come from other cells. So a cell can't just magically appear out of nowhere. A cell has to divide in order to make another cell. So here's those 3 points, I guess written out slightly differently, but in a diagram form, if that's helpful. But those are the 3 points that you want to remember when it comes to what's called cell theory. And this is one of the major theories in biology. Okay. So the rest of the cellular information that I will cover is based on these 3 points. We're just assuming and taking these as really well-supported points in biology. Okay, so let's jump into actually looking at some cells. Have you guys heard of prokaryotes and eukaryotes? At least heard the terms. Okay. So there are 2 major categories of cells. The prokaryotes are the really simple ones. They are less highly evolved is a way to think of it, and they're single-celled organisms. Prokaryotes are single-celled. Eukaryotes are going to be most of what you think about. Most animals that you think about are made up of eukaryotic cells. So these are much more highly adapted and evolved. They have a lot of different organelles, so they can do a lot of things more precisely than prokaryotes can do. Prokaryotes are a lot simpler. We're going to spend most of our time talking about eukaryotes. If you're interested in like microbiology, you'll spend a lot of time looking at prokaryotes, which have some differences, and I'm going to point those out right now. So prokaryotes, the simpler ones, the single cell, you can think of bacteria when you think of prokaryotes. Bacteria are prokaryotes. They don't have a nucleus, whereas eukaryotic cells do. The nucleus is where all the hereditary information, all the DNA is found in a eukaryote. And we'll talk about each of these organelles later. We won't get to it today, but we'll talk about each of the organelles that are found in eukaryotes and what they do. So prokaryotes don't have a nucleus, but they have something called a nucleoid. It's like a general area where the DNA is found, but not a formal nucleus. They do have ribosomes, which is something that prokaryotes and eukaryotes have in common. So eukaryotes have ribosomes, prokaryotes have ribosomes. Those make proteins. So we'll talk about that, but those are the protein organelles. And then, so there is some kind of cell membrane. There's a cell membrane in both. Like I said, we'll talk a lot about the cell membrane, which is kind of a more, I guess, it's flimsier than the cell wall is a good way to say it. So the cell wall is mostly only in prokaryotes and plants. So a cell wall is really rigid. The cell membrane is kind of a little bit more goopy and fluid, I guess is a way to say it, not as solid. Most eukaryotic cells only have a cell membrane. We don't have a wall. Plants do. That's why they can actually stay upright. All right. Those are the main differences. Don't worry about the capsule. You guys don't need to worry about what a capsule is, but know those main differences and then the similarities, the ribosomes and the cell membrane. Those are both present in both prokaryotes and eukaryotes. So I'll point out some other differences between prokaryotes and eukaryotes as we go along. I'll bring prokaryotes up every now and then and say, this is how they're different from eukaryotes. But like I said, most of the time I'll spend talking about eukaryotes. Okay. Cells, individual cells are limited in their size. And this limitation is basically due to a mathematical fact. So I'm going to try to break this down so you can understand it. And then I have a handout somewhere that I want you guys to work on. So for you guys on Zoom, there will be a handout that we'll get to in just a minute. It looks something like that. It should say surface area to volume ratio worksheet. So if you want to go ahead and download that, you can, but we're not going to start on it just yet. Okay, so what is surface area to volume ratio? That just means how much surface area is there versus how much volume of the cell. So surface area is just exactly as it sounds. So how much area is on the outside? And using a cube is an easy way to kind of look at this. Cells are not necessarily cubicle shaped, but how much surface area is there versus how much volume is inside. The important difference here is that area is two-dimensional, volume is three-dimensional. So when we measure area on a cube, we just take the sides and multiply them. And that's going to give us a two-dimensional area. When you're looking at volume, you're looking at 3 dimensions. So you're cubing a certain number. So that means that volume is going to grow a lot faster than surface area. So for each, say, millimeter increase on the outside of one of the edges of these cubes, the volume is going to increase basically another point beyond that. So here, if we look at these 3, well, I don't have the calculations for that, but these 2 cubes, so we'll imagine these are cells. The surface area here is going to be 6 millimeters squared. So that's length times width times height. So I think I have that on the worksheet. Yeah. I'm going to hand out this worksheet right now, actually. So you guys have this in front of you. Might be helpful. Okay, so as you can see on that worksheet, so if you guys want to, on Zoom, if you want to open this up, the worksheet, surface area to volume ratio. The surface area of a cube, you'll see, is 6 times the side times side length, basically. So 6 because there's 6 surfaces of a cube, right? If you imagine a cube, it has 4 sides, and then 1 on top and 1 on the bottom. So that's going to be 6 surfaces around a cube. So again, we're going to assume that our cells, for this purpose, are cubicle shaped. So that's where the 6 comes from. And then the volume is going to be the side times side times side. So I misspoke there when I was describing length times width times height. So this surface area of this cube is 6 times, what was it? Just side times side. 6 times 1 times 1. So 6 millimeters squared. But the volume is 1 times 1 times 1, right? So that's 1 millimeter cubed. So that's where we're starting. So for you guys on Zoom, 6 times basically 1 times 1 is still just 6. And then here, the surface area is going to be 6 times 2 times 2. So 6 times 2 is 12. 12 times 2 is 24. So surface area is 24 millimeters squared, which you see here. And it doesn't matter which sides you choose because in a cube, they're all the same length, right? So just choose 2 of them. But the volume now is going to be 2 times 2 times 2. So 2 times 2 is 4. 4 times 2 is 8. So 8 millimeters cubed. So we went from, this is the important point, when you look at what you jump from in surface area to volume. So surface area, you're jumping from 6 in this cube to 24. So that's a 4 times increase. 6 times 4 is 24. volume, you're jumping from 1 to 8. That's an 8 times increase. Does that make sense? So 6 to 24 is a smaller increase from a ratio relative perspective than 1 to 8. So surface area doesn't increase as quickly as volume does. Can anyone venture a guess as to why the heck that matters? Who cares? So think about the cell membrane and the amount of like volume and stuff inside a cell. So if stuff needs to get across the cell membrane, like get more nutrients into the cell or kick waste out of the cell, if there's a huge volume inside, but not really a whole lot of surface area, what's that going to do? Anyone? You don't have a lot of area to move stuff across, but you have a huge volume. That's going to be difficult to sustain a cell. So a large volume means you need a lot of nutrients coming into that cell. You have a lot of waste that you need to get out. If there's not enough surface area to do that, then the cell can't survive. It can't just keep growing because there's not enough area to bring more nutrients in or kick waste out. So that's why this matters. A cell can only grow so big before it can't sustain itself because there's not enough surface area to move materials across. So make sure you understand that. I'll probably ask you to explain why it matters, in addition to being able to tell me what ratio increase occurs. So I'm going to let you guys for a second work on this worksheet. So the worksheet basically, so you guys have it in front of you. I'll hold it up to you guys. You're going to look at different cube sizes, calculate the surface area and the volume, and then the ratio. So the ratio is just the number that you get for surface area versus the number you get for volume. So you see the example on the first line, and you'll be able to see as the cube grows, how much faster the volume is growing versus the surface area. And then there's some questions at the bottom that you'll answer. So take about 4 or 5 minutes to work on this. And this will be due on Friday before class. So there's an assignment on Canvas. So make sure you upload it to the assignment on Canvas by noon on Friday. And just kind of work on it individually right now and let me know if you have questions. It's a good way to kind of break down the simple math. And then the questions at the bottom make you kind of think about what that math means. just going to work out one on the board to make sure you guys have the hang of this. All right, so hopefully you guys on Zoom can see this. So sorry, I didn't see the chat, Sophia. Yeah, good question, Sophia. And we can talk about this more on Friday when everyone gets it turned in. But yeah, you're on the right track. Okay, so I'm going to stop sharing, and in the last few minutes here, we're just going to work through one example, hoping this marker works, hoping you guys can see on Zoom. Okay, so let's do the second line, which I assume everyone has done by now. So the surface area, the side length is 2, right? So the surface area is going to be, let's see, okay, doesn't see there. Surface area is going to be, of course, and I just have to say this, 2 times 2 times 2, which you can't really, I'm sorry, 6 times 2 times 2, which you can't really, fine. 6 times 2 times 2 is going to be that first block, right? Does everyone have that? Okay, so 6 times the 2 side lengths. So what is that? 24, right? So our surface area is 24 centimeters squared. The volume is 2 times 2 times 2. So just all 3 sides multiplied. And what is that? 8. Yep, 8 cubic centimeters. So what would that make the surface area to volume ratio? 24 to 8, and you can reduce that. So it's just like a fraction. So 8 goes into both of those, right? So it would be, yep, 3 to 1, exactly. So if you divide 24 by 8, you get 3. If you divide 8 by 8, you get 1. So the easiest way to compare these surface area to volume ratios is really to reduce them down. So 6 to 1 was our first one with the smaller cube. 3 to 1 is the second one with the slightly larger sides. So it's an overall kind of a smaller ratio. And the efficiency of the cell, so that's what they're using. They're just dividing, they're taking that ratio, basically 6 divided by 1 for the first one, or 3 divided by 1, which is going to be 3, right? So you should get 3 for the efficiency of that second cell. So that's a really nice way to look at how efficiency actually goes down. As more volume is in the cell than there is surface area. So you're kind of expanding the inside too fast, and the outside is not expanding fast enough. Okay, so complete that, and we'll talk about it on Friday. So you can bring a copy with you, I guess, since you guys will actually still be in here. So bring your finished copy, upload it to Canvas still so I can grade it. But just so you have a copy to look at, bring it and answer those questions on the bottom. We'll talk about that on Friday. Oh, and I'll take your exams as you go out. You can just set them up here.